Here are 5 USMLE-style questions focused on protein synthesis, including initiation, elongation, and termination. Hasher question 1. Which of the following is the primary role of eukaryotic initiation factors, EIFs, during the initiation phase of protein synthesis? A. They recognize the Shine-Dalgarno sequence in prokaryotic mRNA. B. They help assemble the 40S ribosomal subunit with the initiator tRNA. C. They catalyze the formation of peptide bonds between amino acids. D. They facilitate the movement of tRNA in the E site. E. They recognize the stop codon and terminate translation. B. They help assemble the 40S ribosomal subunit with the initiator tRNA. Explanation: Eukaryotic initiation factors, EIFs, play a crucial role in the initiation phase of protein synthesis by recognizing the 5A cap on the mRNA and helping assemble the 40S ribosomal subunit with the initiator tRNA. This process is necessary for the formation of the 80S ribosome that begins translation. Question 2. In eukaryotic translation, which of the following is required for the translocation step of protein synthesis? A. ATP, B. GTP, C. CTP, D. tRNA, E. mRNA. Answer: B. Explanation: GTP is required for translocation in protein synthesis. During elongation factors bind to GTP, allowing the ribosome to move along the mRNA through each direction after peptide bond formation, thereby shifting the tRNA into the P site and E site. Question 3. Which of the following bacterial toxins inhibits protein synthesis by targeting the elongation factor EFs? A. Diphtheria toxin. B. Tetanus toxin. C. Botulinum toxin. D. Cholera toxin. E. Shiga toxin. Answer A. Diphtheria toxin. Explanation: Diphtheria toxin targets the elongation factor, EF, in eukaryotes, particularly EF2, inhibiting protein synthesis by preventing translocation of the ribosome along the mRNA. This leads to cell death and is a key feature of diphtheria. Question 4. In prokaryotes, the Shine-Dalgarno sequence is crucial for initiating protein synthesis. What is the role of the Shine-Dalgarno sequence? A. It binds to the EIFs to initiate translation. B. It aligns the ribosome with the start codon via recognition by the 16S RNA. C. It promotes the release of tRNA from the ribosome. D. It recognizes the stop codon for translation termination. E. It catalyzes peptide bond formation between amino acids. Answer B. It aligns the ribosome with the start codon via recognition by the 16S RNA. Explanation: In prokaryotes, the Shine-Dalgarno sequence is a ribosomal binding site that helps align the ribosome with the start codon on the mRNA. The 16S RNA of the 30S ribosomal subunit recognizes and binds to this sequence, ensuring proper initiation of translation. Question 5. During eukaryotic translation termination, which of the following is required for the release of the completed polypeptide from the ribosome? A. ATP, B. GTP, C. Aminoacyl tRNA, D. mRNA. E. Release factors, ERFs. Release factors. Explanation: In eukaryotes, release factors recognize the stop codon and facilitate the termination of translation. They bind to the ribosome, causing the release of the completed polypeptide from the ribosome. This process requires GTP as well to provide energy for the release. These questions cover key concepts in protein synthesis, including the roles of initiation factors, elongation